Hello everyone and welcome back to Neuropsychology. So chapter 5 is a little bit shorter. This is already the last video for chapter 5. So we have covered a lot in the last video. We talked about neurotransmitters and the structure of a synapse, how neurons convey information from one cell to another, aka through neurotransmitters, and also the different steps in neurotransmission. So in this video, we are going to talk about different types of synapses. So we're going to talk about the excitatory and inhibitory synapses and all the differences between them. We're going to talk about criteria for identifying a neurotransmitter, the 3 classes of neurotransmitters, and I'll give you guys some examples, but not all, because then we're going to be here forever. We're also going to talk about 2 types of receptors, so the ionotropic and the metabotropic receptors. We're briefly going to talk about the 4 activating systems, and then we're briefly going to talk about sudden infant death syndrome, or SIDS, and also sleep apnea. Okay, so there are many different types of synapses, as you guys know. So some neurons synapse with the dendrite of another neuron, like we learned initially, and some neurons synapse on muscle, which we call axomuscular synapses. Some neurons can also synapse on tiny blood vessels, and these will secrete their neurotransmitters directly into the blood. So there are many more different types, but despite all the different types, in the end the synapse can only convey one of 2 messages. So it can give an excitatory message and make the neuron more likely to fire, or an inhibitory message. So these 2 synapses, they actually differ in location and also in appearance. So let's start off with the excitatory synapse, or we also call that type 1 synapse. So type 1 synapses, they're typically located on the shaft by the dendrites, so here on the shaft, or right by the dendrites. So as you can see, the vesicles inside here that contain all the neurotransmitters, they are a little bit more round. So in the other ones, in the inhibitory synapses, they're a little bit more flat. But here, they're very round, and there are many of them. So here you can also see that the cleft, or the area between the 2 neurons, is a little bit wider. So it has a wider cleft. And the active zone is larger. So you can see that there's way more or a lot of receptors here. So also there's dense material in the membrane, so a lot of neurotransmitters will go out and diffuse over to the other side. Okay, so let's look at the inhibitory synapses. So inhibitory synapses are also called type 2 synapses. So these typically synapse on the cell body. So I know we initially learned that a synapse goes to the dendrite. I still want you guys to think about 2 neurons connecting in that way, but just know that in real life they can connect in many different ways. Inhibitory synapses, they synapse most often on the cell body. So the vesicles in an inhibitory synapse are a little bit more flat, so they're kind of more like pancakey. And the synaptic cleft, which is the area between the 2 cells, is way more narrow, so they're closer together. And lastly, these inhibitory synapses, they have smaller active zones, so they have way less receptors in their membrane. And also you can see that less of the vesicles are fusing, so less neurotransmitters are going to diffuse over to the next cell. Okay, so next we are going to talk about how you can identify a chemical to be a neurotransmitter. And then after this, we're going to talk about the categories of neurotransmitters. So there are 4 main criteria for identifying a neurotransmitter. So the first thing is that the chemical has to be created in the neuron. The second thing is when the neuron is active, the chemical must be released and produce a response in some target cell. The third criteria is that the same response or receptor action must occur when this chemical is experimentally placed on the receptor. So if you place, in the picture you see that right here, if you place this chemical on the receptor, it should have a similar effect. And the last thing is whenever the neurotransmitter did its job and connected to or binded to the receptor, it has to be either taken back up in the cell or taken away, or it is broken down. So, by systematically applying all these criteria, researchers can determine which of the thousands of little chemicals or chemical molecules inside of a cell are neurotransmitters. So, why is this important? This is important for example, research, or disease research or drug research, or also when people want to create new medications, they need to know what they have to create to work on these systems. Okay, so let's break up all the neurotransmitters. So we're not going to talk about all of them because there's many of them. But we can broadly divide all the neurotransmitters up in 3 groups. So we have the small molecule transmitters, the peptide transmitters, and the transmitter gases. So let's start off with the first one. So the first group are quick-acting small molecule transmitters, and an example of that is acetylcholine. So typically they're created from food or your dietary nutrients, and they are packaged for use in your axon terminals. So whenever a small molecule transmitter is released from an axon, it can quickly be replaced at the presynaptic membrane. So because they're stored by the axon terminal, this is why they're very quickly working. So since the main component of this group of neurotransmitters comes from our food, our diet is really, really important. So our diet has a big influence of how many of these neurotransmitters are in our brains or in our bodies. So keep in mind, always eat healthy. So within the small molecule neurotransmitters, we have 2 other groups. We have the amines, as you can see right here, which includes dopamine, noradrenaline, and adrenaline, and serotonin. And you have the amino acids. So let's go over those in a tiny bit more detail. So amines are a subgroup of the small molecule transmitters, as we just said. These are chemicals that contain an amine group, which, if you've had chemistry before, this is the NH group in their chemical structure. Some amine transmitters are synthesized by a common biological pathway, and they're related. So an example of that is dopamine, noradrenaline, and adrenaline are related, and here is their pathway. So from dopamine, using an enzyme, you can make noradrenaline, and from noradrenaline, using another enzyme, you can make adrenaline. The precursors of these are L-DOPA and tyrosine. So an example of food that has a lot of tyrosine is, for example, harder cheeses, or also bananas. So in Parkinson's disease, there is an insufficiency of dopamine. So you cannot give someone directly dopamine because it will not pass through the blood-brain barrier that we talked about earlier. However, L-DOPA does pass through it. So often L-DOPA is used to increase dopamine levels in people that have Parkinson's disease. Okay, so these were all examples of small molecule transmitters. Serotonin is a little bit differently. It does not share a common path with the ones that I named before. But serotonin is very important for mood and aggression regulation. It's also important for appetite and arousal, pain perception and respiration. And it comes from the amino acid tryptophan, which you've definitely heard from. You can find that in turkey. You can also find that in milk or bananas. Okay, so the next group are the peptide transmitters. So neuropeptides are technically multifunctional chains of amino acids, and they act as neurotransmitters. So they're kind of similar as proteins. So these are made through a similar pathway as proteins. They're made through translation of messenger RNA, which, if you remember, it is a copy of a part of a DNA strand. If you don't remember this, go back to the transcription and translation video. So most of these are assembled just like proteins in the ribosomes and then packaged by the Golgi bodies and transported to the axon terminals. So they have a huge variety of functions. I only put a couple here, but they have more functions than this. So they can, for example, serve as hormones. An example of that is the growth hormone. They are also active in response to stress, for example, corticotropin. They facilitate learning. They can regulate your eating or your drinking. So I know vasopressin is important for that. And respond to pleasure or pain. So an example of that are your beta endorphins. So don't worry about the names of the neuropeptides that I just mentioned. I just want you to know that neuropeptides can serve in all these functions. Okay, and lastly, we have the transmitter gases. So water-soluble nitric oxide or carbon monoxide can both act as neurotransmitters. So they are synthesized by the cell whenever the cell needs it because they cannot be stored. And unlike the other two groups, these can be produced in many regions of the cell. So nitric oxide serves as a messenger in many parts of the body. It has a variety of functions. So examples of it is it controls the muscles of your intestinal walls, it dilates your blood vessels and brain regions that are in active use so that these regions get more blood. And it also dilates your blood vessels in genital organs, and therefore it is active in producing erections in males. So there's a drug called sildenafil citrate, or I'm sure everyone has heard of it as Viagra. And Viagra enhances the action of nitric oxide and is also used as a treatment for male erectile disorders. Okay, so now we talked about the different groups of neurotransmitters. Let's talk about the two types of receptors. So whenever a neurotransmitter is released from the presynaptic membrane, it diffuses over from one cell to the next, and then it binds to a receptor on the postsynaptic membrane. However, what happens next depends on the receptor, and there are two types of receptors. We have the ionotropic receptors and the metabotropic receptors. The best way to differentiate these two are to think about the ionotropic receptor as the simple ones and the metabotropic receptors as the complicated ones. So ionotropic receptors technically just have two parts. They have a binding site for a neurotransmitter, and they have a pore or a channel, just like we went over before. I'll show you guys a picture in the next two slides. So this pore can regulate the movement of charged atoms across a cell membrane whenever a neurotransmitter binds to the binding site of this receptor. So ionotropic receptors are usually excitatory and increase the likelihood that the neuron will produce an action potential, so it gives an EPSP. Ionotropic receptors can also change the membrane voltage very quickly. This is because they're pretty simple, they can open and close pretty quickly. However, because of this, their effect does not last very long. So metabotropic receptors are a little bit more complicated. So you'll see it in two slides further. So these receptors are usually inhibitory and decrease the likelihood that a neuron will produce an action potential. So what do we call this again? Do you remember? Okay, so if you remembered, it is an IPSB. So this receptor also has multiple parts, but it has more parts than the ionotropic receptor. So first you have the outer part, which is the part that binds with the neurotransmitter. So this sticks out of the membrane and has a little area where the neurotransmitter can attach to. Then the internal part of the receptor is associated with what we call a G protein. So this will translate the message from the neurotransmitter in some sort of biochemical activity within the cell. So the G protein has 3 subunits. It has an alpha, a beta, and a gamma. So technically what this system does is whenever a neurotransmitter binds to it, there's another part, a second messenger, that will carry the message to other structures within the cell. So since this receptor is a little bit more complicated, it is also a little bit slower. However, the effects are longer, they last longer, and the effects are indirect. Okay, so let's look at these to make a little bit more sense of this. So here you see an ionotropic receptor. So ionotropic receptors has two parts. So it has a binding site, as you can see right here. And then it has a pore where molecules or chemicals can go through. So the pore regulates the movement of charged atoms across the cell membrane when a neurotransmitter binds to the binding site. So whenever these little neurotransmitters in red bind here, this can open up and then these ions can go through. So because of the binding of the transmitter to the receptor, it is quickly followed by the opening or closing of the pore. So this is why they work a little bit quicker. So again, in red you have the neurotransmitter, they bind to the ionotropic receptor, the receptor opens and these ions can go through. Take note that the neurotransmitters do not go in the cell. All they do is deliver a message, let the protein do whatever it needs to do, and then they go back into the cleft. Okay, so here you see a metabotropic receptor. So there are two different types of effects it can have whenever a neurotransmitter binds to it. So here you see the receptor, and here you see, for example, a channel. And on this side you see the receptor right here, and here you see an enzyme. So in the first one, for example, whenever the neurotransmitter binds to the metabotropic receptor, then eventually the G protein is activated, the alpha subunit will detach, that will attach to a pore or a channel, and then the channel will open and let atoms or ions in. So this is one way. And another thing it can do is whenever a transmitter binds to a metabotropic receptor, then the G protein gets activated, the alpha subunit detaches, it can attach to an enzyme, so this is an enzyme, and then an enzyme can do a lot of different things. So what we call this is then the second messenger. This sends an information to the second messenger, and the second messenger carries the message to other structures within the cell. So the second messenger can, for example, bind to a membrane channel, causing it to open or close. It can also initiate a reaction that could, for example, create more ion channels. And it can even influence the DNA of our cell to initiate or cease gene expression. And technically, therefore, indirectly, it can regulate protein formation. Okay, so I know the metabotropic receptor is a little bit difficult to understand. So just pause the video and take some time to look at these images. And once you understand the images, move on to the rest of the video. Okay, so now we've covered neurotransmitters and receptors. Let's move on and briefly talk about the neurotransmitter activating system that are important for our behavior. So although many of the neurotransmitters in our brain and spinal cord have very specific functions, others appear to have a very widespread modulatory role of neuroactivity in the central nervous system. So we call these systems activating systems, and they form neural pathways that coordinate brain activity through a single neurotransmitter. So these systems have been implicating in controlling functions such as motor behavior or arousal and mood. And we're going to talk about the four major ones. So we have the cholinergic, dopaminergic, noradrenergic, and serotonergic system. And let's go through these very briefly, but in a little bit more detail. Okay, so let's start off with the cholinergic system. So this system plays a role in normal waking behavior, and it's also important for attention and memory. So people with Alzheimer's disease have a loss of cholinergic neurons during autopsy. So whenever they looked at their brain after they died, they saw that there were less cholinergic neurons. So the brain abnormalities associated with Alzheimer's are not limited to cholinergic neuron loss though, so keep that in mind. Then we have the dopaminergic system, and this one has two pathways. So here you can see it in orange and in purple. So let's start with the first one, which is the nigrostriatal pathway. And this starts in the substantia nigra, so it's orange in this picture. And this system is important for coordinating movement. So when dopamine neurons are lost from the substantia nigra, you have trouble coordinating your movement. And that occurs in Parkinson's disease that we already talked about. So the other pathway is the mesolimbic pathway, and this one starts in the ventral tegmentum, and it is purple in this picture. So this system is often affected in addiction for any type of addiction, so an addiction to food or drugs, or other addictive behaviors such as gambling. So the mesolimbic system enhances responses to environmental stimuli and technically makes them more rewarding. So excessive activity here has also been proposed to play a role in schizophrenia, in which behavioral disorder, the characteristics of this are delusions or hallucinations, agitation, and there are many more characteristics for this. Okay, so the third system is the noradrenergic system. Behaviors and disorders related to the system are related to emotions. So, for example, major depression could have a relation with a decrease in noradrenergic neurons. And mania might also be related to an increase in noradrenergic neurons, and decreased noradrenaline has also been associated with attention deficit hyperactivity disorder. And lastly, we are going to cover the serotonergic system very briefly. So this system is involved in wakefulness as well, and it's also important for learning and memory. So decreases in the system are related to depression, and increases in the system are related to a couple of disorders, including sudden infant death syndrome. So let's talk about SIDS a little bit more. So SIDS, or a sudden infant death syndrome, it's very sad. It's a sudden unexplained death of a healthy infant under a year old, and it often happens whenever the baby goes to sleep. They just stop breathing. They don't feel it. They just stop breathing. So researchers have observed abnormalities in the brainstem, and infants who die of SIDS often show a reduction of serotonin neurons. So there's also an abnormality in serotonin neurons in people that have sleep apnea. So the same brainstem system may also be related to sleep apnea. So sleep apnea is when you have impairments of breathing during sleep. So again, there are abnormalities in your serotonin neurons for sleep apnea. Okay, so this was the last slide, and this is the end of the video. So I will see you guys back soon for the next one.